Welcome everybody. Good afternoon. Jason and I are very excited to be here today to talk to you about a couple of new initiatives we've had on the DataSpace this year. First thing I'm going to do is just give you a little introduction to the DataSpace, and then we're going to talk about those two initiatives. I'm going to go over the first one, which is where we expanded the membership to the public, and then Jason's going to talk about where we brought in some new data types, monoclonal antibody data, not unlike what Kira was just talking about. Am I getting feedback, or is this mic okay? Okay, great. So let's get started. First, just a little bit of a flash from the past, perhaps. For those of you who may have been to a few of these user conferences, you may have been at the 2014 LabKey user conference, where Britt Peeler and I presented on a project we were working on called the Collaborative DataSpace. And this was a program that was designed specifically for HIV vaccine research. And it's really designed to be an integrated program with 4 primary components. And the first component is, of course, the data from these HIV vaccine trials. Not only integrated data across all the subjects for all the studies, but also metadata about those studies and the products that are being tested in them. We talked about another component for services, that's outreach, analysis support, and general support, and then governance. This is to facilitate collaboration across the members. It's for data sharing conventions and guidelines, and then practices around harmonization. And then the fourth component was around the application, and this is specifically a web-based LabKey, using LabKey software, web-based application that allows the users to explore the data, do some visualizations, and then export that data. So what we talked about in 2014 is that for us, it's very important for this program that these 4 components be tightly integrated and working together. And we felt like that's going to mean success for the program. And we believe that firmly today. In 2015, we launched this program for one of our network partners, the Collaboration for AIDS Vaccine Discovery. It's a Gates-funded network. And so we launched this whole program for the CAVD, and we branded the application portion as the CAVD DataSpace, and that's what we're going to be talking about today. So the CAVD DataSpace is a data sharing and discovery tool that we've developed for the CAVD, but also for the broader HIV vaccine community. We developed it not only to enable and even encourage the exploration of the data beyond the primary analyses that were in the publications, but also we aim to make the community more aware of the research that's being done across the field and to facilitate collaboration amongst researchers. Our primary target audience for the DataSpace are the researchers that are involved in concept development, study design, and ancillary studies. So this could be principal investigators who have their own funding, their own studies, but it could also be junior investigators like staff scientists, postdocs, who may not have their own funding and studies and are just starting their careers and looking for data to use. It's also targeting now statisticians and other people in data science and bioinformatics that are looking for data to explore further. So there's 3 main things that users can do in the DataSpace. The first is to answer questions about studies. So what were the products used? What were the assays tested? The second thing that folks can do is to find a cohort. So this would be locating subjects with common characteristics, like all having received a similar product that can be compared. And then the third is being able to explore the data and the relationships across those studies and data. So currently in the DataSpace, because our main funder is the Collaboration for AIDS Vaccine Discovery, we are focused right now on CAVD studies. So the first thing that we have in there is study information for all the completed CAVD studies. And just to give you a sense of what I mean by that, so for each of our studies that we show in the DataSpace, we have what we call a study page in our, we call it the Learn About section. And so there's a study page for each of the studies, or might call them experiments as well. And so these pages would include general study information, so what type of study is it, what were the species that were being tested, and what's the status of that study. It would also include information like a description of the study, what were the study objectives, what were they doing the study for, rationale, a description of the treatment groups that are being tested, what products they received and at what time, additional contact information for follow-up questions. We list the products that were being tested in the study. We list the assays that were tested in the study, including whether the data is available for those assays. And then there's a section for additional reports or other documents to support that study. And then we'd also include, if you were to scroll down, you'd see conclusions and even links to publications. So we have a similar page for our vaccine products that talk about general product information. Is it a vaccine? Is it an adjuvant? What's the class? Is it a DNA? Is it a protein? A brief description. We show the studies that test that particular product, and then any other products that have been tested along with that product. And then we have a similar page for, yes?
Sorry, could we just get the speakers turned up? No,
Okay. Are you on that back there? Okay. Is this better?
Yes.
Okay. Yeah, so we have a similar page for all of our assay data that we have in there that describes the assay being tested. So along with the information about the studies and the products, we have included integrated data for each of the assays for the completed studies. So this includes assay data for 3 assays at the moment, binding antibody multiplex assay, the BAMA assay, intracellular cytokine staining, and a neutralization assay. And we do also have a little bit of ELISA data, or ELISpot data. So this data is from human clinical trials, non-human primate trials, as well as other animal trials. And then recently, we've begun adding monoclonal antibody study data as well. And Jason's going to talk about that a little bit in a moment. We've also started working with one of our other HIV networks to process some of their data, and we hope to be adding that in soon. So this integration of study data adds a lot of value to the assay data. By standardizing this data across the studies, we're really facilitating meta-analyses and comparisons across the studies. Some of the standardization that we've been applying is a common data structure and field names across the studies. We apply a consistent timing variable in study day. So some of our studies will come in, they're date-based, others come in and they're visit-based, and we will convert all of those to a study day, give us a common frame of reference as we're looking at the data. We'll also do coding of things like viruses. One lab may use a different name for a virus than another lab, and so we've maintained a master list, and we will code to that master name so it's all consistent. We apply additional metadata about those viruses. And then we also apply other controlled terminology for assay dimensions. So maybe target cells are always called the same thing, or we use consistent naming for antigens. And then for some of our assays, we do an additional calculation on a response call. So if there's been a response call calculation that's changed over time, we do a recalculation so it's consistent across all the studies. We'll also add in some additional fields that may not have existed in the analysis data sets. So we use publication IDs instead of participant IDs to further de-identify the human subjects. We'll include some additional measures like area under the curve or a titer at a specific concentration that may not have been in the data set originally. And then we bring in additional assay metadata like specimen type, what was the name of the lab that conducted it, whether the antigen matches the vaccine that's being tested, et cetera. So all this standardization makes it possible for to easily view the data across studies. So what we're seeing here is one of the sections, we talked about some of our pages in Find. All these other areas deal with the data directly. So this is one area in the application that we call plot. What we have here is a simple plot of the MAb titer, the neutralizing antibodies titer, for 2 of our studies. And what you can see with these little syringe icons, those indicate when the vaccinations were administered to the subjects. So you can see that the different groups can have different vaccine regimens and the timing of those vaccines. So this gives you a quick view of how those compare. And what I've done in this particular view is that I've been able to realign all these time points by the last vaccination that was administered. That gives us a quick view of the peak immunogenicity time point. So I can grab this, filter this out, and do further looks at that. So that's one of the really powerful things of our application, is being able to realign those time points. Okay, so who has access to the DataSpace? Well, CAVD paid for it, so for sure their members have access, so current and former members of the CAVD, as well as invited members from some of the other HIV vaccine networks, like the HIV Vaccine Trials Network. We've also got affiliates that have been invited by the foundation for some of their other disease programs that do vaccines as well, like their TB and polio programs. And then as of July, we opened it to the public. Which leads me to the first initiative, which is our expansion of membership to the public. So why did we do this? Well, the goal of the DataSpace is really to be a resource for the broader HIV research community. One of the things that we realized early on was that a lot of the information and data from these studies was siloed within the networks. So if you're not a member of one of those networks, you don't have good visibility into the activities, and you certainly can't get your hands on the data very easily from a network if you're not a member. That's compounded by the fact that network membership can be transient. So some of these networks, your participation in that network is contingent on the fact that you have an active award. So you may have an active award, you have good visibility, you're finding out what's going on, but if your award is not continuing, or if you're in between active awards, you kind of lose that visibility. The other point is that not all research is conducted within one of these networks. So there's a lot of people out there doing their own research, either with smaller organizations or on their own, and we wanted this information to be accessible to all of those. So by expanding the membership to include beyond those networks, we feel like that gives the research community a more timely and complete view of activities. That's going to alert researchers to studies that may be relevant to their work. It's going to encourage more collaboration. It allows researchers to plan for ancillary studies and data analyses ahead of time, especially if that's going to require funding. And then it reduces duplication of effort across projects and awards. So if an investigator is preparing an application for funding for a study, and they identify a similar study or the same study that's already been conducted, this gives them the opportunity to make adjustments to that application. Some of the considerations that we thought about as we were planning this public access. The main one is that not all the data is approved for sharing. We have in the CAVD, it's largely animal trials and NHP trials, but we do have human trials as well. And as we get into the HIV vaccine trials networks, that's entirely clinical trials. So you have clinical trial agreements and consent forms that can dictate who can see certain data. And there are also network data sharing policies that can specify what data can be shared as well. So we needed a way to be able to customize the access to the data by study. We also wanted to be able to indicate to the users when the data was restricted versus accessible to them. And then finally, what we wanted to be able to do with the study pages is to make sure that those were accessible to everyone. We felt like that was really important to give that broad, comprehensive view of the activities that are going on. So we didn't want those to be restricted. And we encourage our networks to make those study pages available before the study is complete, which means that there can sometimes be information that we might be on those pages that would be considered sensitive prior to the study being complete. So there are 4 main areas that we worked on during implementation. The first is what we call a staged collaboration level. The second is a data availability flags. We set up a self-registration process, and then we've done some updating on our data sharing and use agreements. And I'm just going to step through those quickly. The staged collaboration levels, what we were trying to do here is set up 3 main levels of access. The first one we called restricted collaboration, and that might be for 1 or more site groups. Maybe it's a specific network or a protocol team. The second is what we call full collaboration, and that would be data that was accessible to all network groups. And then finally, the public access, and that would be accessible to all site users. So what we did to create this was we used the site groups, and we nested those site groups to create the staged collaboration levels. So if you're someone who has administrative responsibilities, you might recognize this particular interface of the user groups. So we created one. You have the individual ones for each network, and then we created a full collaboration and then added each of those individual network site groups within that full collaboration. What we were then able to do is use that nested group to define the permissions for each study. So you've got your study folders over here. There's one for each study, and we can then define for each of the studies what the access to that study data should be. And then lastly, of course, assigning each of the users to 1 or more site groups as appropriate. So if it's a member of the CAVD or a member of another network, then they could go into 1 or more of those site groups. Data availability flags. So the point here was to be able to indicate to the users when data is accessible to them and when it's not, and ideally why. So we identified 3 main flags that we wanted to set. And this area shows up on the study pages and in a few other areas of the app where we're indicating when data is available. But essentially what we've done is to give a quick visualization. The green check mark indicates that the data has been loaded and is available to them. The grayed out check mark indicates that the data has been loaded, but it is not accessible to them. And then the gray X indicates that the data has not yet been loaded. And then you can't see this on the slide, but within the application, if you were to hover over that, you would get a tooltip that would tell you what the status was and maybe why it's not being shared. We wanted the users to be able to quickly and easily log in on their own, so we developed a registration process. This is our public page for the DataSpace, and there's a registration button here. They go in, give us some quick information, and they are logged in pretty quickly. So we wanted to balance that with knowing who people were. So we did want to know who was accessing things, and we do a lot of metrics on who's in there and what they're doing. So we developed a little registration, I think it's probably a user survey is what it is, and get some basic contact information, their institutional affiliations, their network affiliations, and then any research areas of interest. And we use this information to check for existing accounts. If they had a network account as well, we'd want to reconcile that and get it down to just one account where it had maximum access. And then we also use it to identify potential outreach opportunities, projects or other collaborations they might be interested in. And then lastly, our data sharing and use agreements. The current agreement references an existing agreement for the CAVD, which is their data and materials sharing agreement, which is specific to the CAVD and its data. As we bring on additional networks, what we will be doing is creating a data governance committee with representation from each of those networks. And we will work with them to establish a DataSpace specific agreement. We'll also want to use that data governance committee to work through some additional topics, including establishing some norms and expectations around data use from data out of the DataSpace. So we want to talk to them about how and when we should restrict access and who gets to decide. Well, should we let the networks decide how they're going to restrict it, or should there be some general conventions that are required? Are there any restrictions on how data should be used? If someone exports the data out of DataSpace, are they allowed to do a publication on it and present on it? Or are there any restrictions on that? How should individual network publication policies be followed? So for a lot of networks, they'll have a review committee that is required to review the publication before it is published. And are those in place when the data is received through the DataSpace, or are there other mechanisms? And then finally, should there be any requirements for citations in the manuscripts, either citing the DataSpace itself, the contributing networks, or the investigators that produce the data? So I think that four items, and those actually went pretty smoothly. And I think that I would contribute that a lot to sort of us at the outset of this project anticipating that we were going to want to have public access. So in the ways that we developed the back end of the DataSpace to begin with, we kind of had that in mind. So that made this extra step pretty smooth. Jesse might have another opinion because they did a lot of the work on the schemas and everything to make it work as well. So as I said, we went public in July, so feel free to log in, dataspace.cavd.org. And our next steps from here is really doing more outreach. This is something, we've focused a lot on the data over the last several years, and I think what we really need to do now is make sure people know about it and know how to use it, and so we have a lot of outreach activities on our plate going forward. So with that, I'm going to turn this over to Jason.
No? Close enough? Dun, 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 yeah.
Green advances?
Thank you.
Okay. It's not a laser guy.
Yeah. Hello, everyone. My name is Jason Taylor. Oh, yes. Trina, thank you. My name is Jason Taylor. I've been on the DataSpace team for about 8 months now. And when I came on, there was an initiative to bring the monoclonal antibody data to the application. And so I've spent a lot of that 8 months munging those data from various sources and bringing them to the app. So I'm going to talk about that. So why are we storing MABs? I know we have a mixed crowd here, and it's been a process for me to understand why would we want to store MABs in the database. I'm going to kind of go over that for a second here. So everyone knows what monoclonal antibodies are? Yes? No, it's mixed. Okay, we're going to go for it. So, monoclonal antibodies are antibodies where we understand the sequence, and they can either be discovered or they can be engineered. We were talking about discovering monoclonals earlier. And for HIV, monoclonal antibodies are considered like a pretty promising technology in regards to prevention and treatment. So traditional methods for making MABs, or monoclonal antibodies, the technology dates back to the 70s, and it was discovered that if you could fuse a mouse spleen cell that was having an immune response to a given antigen with the myeloma cell, a cancerous B cell, you could create these hybridoma cells that would in turn crank out a particular type of antibody. You could select for those cells, and then you would have this immortal cell line that would make a particular antibody that you could study and understand. So the technology has advanced quite a bit since then, and using murine or mouse-based antibodies for treatment and prevention can cause problems with immunogenicity to the antibodies themselves. And you can make human hybridomas, but that technology has been, as I have read, has been traditionally inefficient and difficult to maintain. But technology has persisted, and there are all kinds of ways that we're making monoclonal antibodies now. So sequencing chimeric and humanized antibodies, you're taking like the sequence from a binding region that you're interested in, and then humanizing it by adding it to a human antibody framework. You have camelid antibodies. These are antibodies that come from a camel-type species, llama, alpaca. They're very unique in the sense that they only have a heavy chain region. And then also you can take antibody genes and you can transfect those into plants and yeast and use those organisms to produce antibodies. They're also looking at producing these antibodies in vivo or within a given organism. So with all that, the CAVD has studies in Atlas that look at some of these modern methods. CAVD 399 looks at these camelid antibody fragments and the neutralizing power of those antibodies. CAV 410 is looking at this particular MAB, VRC01, and transfecting those antibody genes in plants, particularly tobacco plants, and mass producing these antibodies as a way to sort of figure out a cost-effective way of producing that particular monoclonal. So how do we bring those data from the CAVD into DataSpace? Well, DataSpace, up until last year, DataSpace was storing neutralizing antibody assays. And it wasn't going to be too difficult to modify that process to bring over the monoclonal antibody neutralizing antibody assays. One of the differences, though, is that these data types are they're different. Where with MAB studies, you're looking at a particular monoclonal and its neutralizing power. In a NAB study, you're looking at a polyclonal serum, and the subject is an actual individual rather than a given monoclonal. So even though the assays are similar, the data themselves are different. Also, with NAB assays, you're looking at dilution of serum and the neutralizing power of various dilutions, whereas with MAB studies, you're looking at the concentration of a given MAB. So concentration and dilution are similar, but they're represented different in the data. So right now, the CAVD has nearly 100 MAB studies sitting in Atlas, which was a LabKey server that was mentioned by Brendan earlier. Yes. So we had tools that were developed for bringing over NAB studies into DataSpace, and it wasn't. We were able to look at those tools and look at those processes and modify those so that we could handle these new sort of idiosyncrasies in the data. So as I mentioned, we had, with NAB studies, they're concentration-based. Excuse me, NAB studies are dilution-based. Yes, NAB studies are dilution-based, but all the NAB data that we have in DataSpace right now just stores a Titer value, a Titer 50 value. And what we wanted is the dilution level data. What we wanted to show to users are those neutralization curves. And so we had to make a modification in our process to account for that. Here's sort of an overview of the process. I think of it in 3 steps. We have the Atlas server that's storing the monoclonal antibody assay data that we want to bring over. And then we've written an R package that we call Atlas MAB Grab, for lack of a better name. And what it does, it goes to Atlas, and it has a bunch of tools that essentially it's making a curl call within R to bring over that JSON and then munge those JSON files into a usable R object. And now we have what comes out of Atlas MAB Data are these very nice, tidy data.table objects. We can bring those objects over into another package, Atlas MAB Data. And this is what we have these a lot with DataSpace and in the lab that I work at, the RG lab, where we have these R data packages. They're essentially R packages that store data. And so when you install the package, you're actually installing data files that you can call very easily. So Atlas MAB Data is one of those packages. And in that package, it's doing a ton of processing. It's calling Atlas MAB Data. It's getting those nice, clean data.table objects into the NGR environment. And then it's cranking out, I think we have 5 quality control reports. And when we're looking at, how many studies do we have at this point? Like 50 or 60? Yeah. It's a lot. We have a lot of studies in there, and each study is going through a quality control report. Drina looks at those quality control reports, and then we're in a tracking CSV. We start marking off the studies that pass various quality control steps. And then this is an iterative process. Finally, studies that pass all of the quality control checks move to another R package called CDS data. And in this package is where we're storing all of the data in DataSpace. From here, from CDS data, files are exported, and an ETL that we have written in Postgres. Not sure. OK. I guess. Good. All right. Doing well. An ETL picks up those files that are exported from R and loads them into DataSpace. So one of the big challenges that we had in that process is MAB name mapping. As you might imagine, when you're pulling data from multiple labs, people aren't necessarily using the same names in the data that they provide people. So we wanted to make sure that we're using the same MAB name, or at least having some kind of track of where labs are calling a MAB a particular name and how we're standardizing those, so that when a user calls for a given MAB, they're going to get all the MABs that they're interested in. So this process, we're going to kind of walk through that a little bit here. You have study A and B, and then you can see here that this is actual data that we had to work with, that we have 4E10, and we've got 4 different versions of that. One of those is probably misspelled. That's what we decided. And so the source name is what the lab gives us. The label name is sort of us just cleaning up that MAB name. The labs thought it was important to denote what source the MAB came from, Polymun or Plantform. These are different companies, I believe. And so we maintain that here in the label. And then we finally say, OK, we know this is all 4E10, so we have this standard name. And then we bring over metadata from the Los Alamos National Laboratory, who has a big repository of monoclonal antibodies that they're tracking. And we bring over that LANL metadata, as well as a LANL ID, so that a user, when they pull those MAB data down, if they want to find that on LANL, they can. And so yeah, we have the data. We know what it is. It's in the app. Now how do we get it? So LabKey helped. We worked with LabKey to develop this thing called the MAB grid. And so Drina showed a screenshot of the app earlier. And so this right sidebar here now has a new section, monoclonal antibodies. You click here, and now you have this nice grid. And this is a summary table of all the data that we have available at the time. And each one of these columns has a filter icon you can see there, and you can sort. So you click on a filter icon. It gives you a list of all your options. You select from that list of options. Then your grid is collapsed based on that selection. And then finally, you select a series of monoclonals in that far right-hand column. And then you can export or generate a report. So the reports, a lot of effort has gone into making really nice reports for this application. And we added these new report buttons at the top. And here's what one of the reports looks like. The actual report that you'll have to sign up and check it out yourselves if you want to see. The actual report that you get actually has tabs. And so when you select multiple monoclonals, you get all of those as an individual tab. And you can click the different tabs and have it and look at a glance at all of the breadth of neutralization for a series of viruses that you see here. Also, we offer heat maps. We'll go back one. This is all done in ggplot2, and it's run as an R report. And this one here is a heat map, and it's made by Heatmap. We might change that to a more lower-level plotting, but it's how it's built for now. And then, yeah, so that's how you're getting data in and out of DataSpace, the GUI. Also, a programmer that we work with, his name's Ju, he has been maintaining DataSpace R, which is a package that you can get off GitHub. DataSpace R is a R API that enables you to query the database or DataSpace directly from R and retrieve R-friendly objects. So getting MAB data using DataSpace R is really straightforward. Once you establish a connection variable, you can just browse a MAB grid by calling the MAB grid method, and then filter rows by calling a filter MAB grid method, and then retrieve your data by just running get MAB. It's very straightforward. What are we looking at here? OK, so getting this connection is just calling connect.ds, and it pulls from a netrc file, I believe, your credentials. And then once you have that connection, you can say connection and call the MAB grid method, and it'll print out to the screen exactly what you would expect to see in the application, that MAB grid, where you have the same columns there. And then once you kind of know what you want, you can. It's a blessing and a curse. OK. Yes, where are we? OK, so once you know what column you want to filter on, you just call that column, and then send it a vector, a character vector of the items that you want. And you can do the same thing with any of the other columns. Once you run that filter, you call the grid again, and you can see your filtered grid. And you can also chain those filters. So this is really cool. Call your connection, call filter MAB grid for this item, call filter MAB grid for the next item. And you can filter it in a really succinct sort of way. If you want to know what if you're looking for all the unique values in a given MAB grid, you can just do that simply with a single command. And then finally, once you have your MAB grid filtered like you want it, then you can just call the get MAB function, and there's your MAB data in a nice R object. There you are. The output is the same as you would expect from the GUI. If you're interested in this package, you're welcome to check it out on GitHub. It's publicly available, and it's well documented. Except for, I should say, the monoclonal antibody is in a feature branch right now. It hasn't been pushed to master. So if you're looking for that, that's where it is. OK. So shall we acknowledge together?
Yeah, sure.
OK.
Yeah, we want to acknowledge all the hard workers on this project, and especially the LabKey group. They have been awesome to work with over these many, many years, and really appreciate all their support. We have Artifact, who is a designer firm locally that we've worked with on the interface, and they've done some great work to make it look real nice. And then our team at the Hutch as well.